Hello, everyone. Today we are going to describe the gram staining question and answer. Question: what is gram staining? Answer: gram staining is a differential staining technique used to classify bacteria into two groups, gram-positive and gram-negative. This classification is based on the structural differences in bacterial cell walls, particularly the thickness of the peptidoglycan layer. Question: describe the steps involved in gram staining. Answer: the gram staining process involves four main steps. 1. Crystal violet, the primary stain that colors all bacteria purple. 2. Iodine treatment. Acts as a mordant, forming a complex with crystal violet, making the dye adhere more strongly to the bacterial cell walls. 3. Decolorization. Alcohol or acetone is used to wash away the stain from gram-negative bacteria, which have a thinner peptidoglycan layer, while gram-positive bacteria retain the stain. 4. Safranin, the counterstain that colors gram-negative bacteria pink or red, while gram-positive bacteria remain purple. Question: what is the primary stain used in gram staining? Answer: the primary stain used in gram staining is crystal violet. Question: what color do gram-positive bacteria appear after gram staining? Answer: gram-positive bacteria appear purple after gram staining. Question: why do gram-positive bacteria retain the crystal violet stain? Answer: gram-positive bacteria have a thick peptidoglycan layer in their cell walls, which traps the crystal violet iodine complex, making them appear purple. Question: what is the role of iodine in gram staining? Answer: iodine acts as a mordant in gram staining. It forms a complex with the crystal violet dye, making it more difficult to wash out of the bacterial cells, especially in gram-positive bacteria. Question: what is the purpose of iodine in the gram staining process? Answer: iodine acts as a mordant, which binds with the crystal violet to form a complex that is more difficult to remove from the cell wall, especially in gram-positive bacteria. Question: what is the role of the decolorizer in gram staining? Answer: the decolorizer, alcohol or acetone, differentiates between gram-positive and gram-negative bacteria by removing the crystal violet stain from gram-negative bacteria. Question: why is the decolorization step critical in gram staining? Answer: decolorization is the critical step that differentiates gram-positive from gram-negative bacteria. Proper decolorization with alcohol or acetone removes the crystal violet from gram-negative bacteria due to their thinner peptidoglycan layer, while gram-positive bacteria retain the dye. Question: why is it important to control the timing of the decolorization step? Answer: over-decolorization can cause gram-positive bacteria to lose the stain and appear gram-negative, while under-decolorization can cause gram-negative bacteria to retain the crystal violet, appearing gram-positive, leading to inaccurate results. Question: what is the counterstain used in gram staining? Answer: the counterstain used in gram staining is safranin. Question: why do gram-negative bacteria appear pink or red? Answer: gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane. During the decolorization step, they lose the crystal violet stain and take up the safranin counterstain, appearing pink or red. Question: what type of bacteria have an outer membrane? Answer: gram-negative bacteria have an outer membrane in addition to a thinner peptidoglycan layer. Question: why is gram staining important in microbiology? Answer: gram staining is important because it helps identify bacteria and determine the appropriate antibiotic treatment. Question: how do gram-positive and gram-negative bacteria differ in gram staining? Answer: gram-positive bacteria have a thick peptidoglycan layer that retains the crystal violet iodine complex, so they appear purple under a microscope. In contrast, gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane that doesn't retain the crystal violet after decolorization, so they take up the safranin counterstain and appear pink or red. Question: what factors can affect the accuracy of gram staining? Answer: Factors that can affect the accuracy of gram staining include the age of the bacterial culture. Older cultures may not stain correctly. The duration and timing of the decolorization step. The quality and concentration of the reagents used. Question: why is gram staining important in clinical diagnostics? Answer: gram staining is crucial in clinical diagnostics because it provides rapid and preliminary identification of bacterial infections. It helps guide the initial choice of antibiotics, as gram-positive and gram-negative bacteria often respond differently to treatments. Question: what would happen if you skip the decolorization step in gram staining? Answer: if the decolorization step is skipped, all bacteria, regardless of their gram status, would appear purple because the crystal violet stain wouldn't be removed from the gram-negative bacteria. This would lead to incorrect classification. Question: what are some limitations of gram staining? Answer: gram staining does not work well on bacteria that lack a cell wall, e.g. mycoplasma, or those with atypical cell walls, e.g. mycobacterium, and it may not provide reliable results if the sample is old or improperly prepared. Question: how does gram staining aid in antibiotic selection? Answer: gram staining helps distinguish between gram-positive and gram-negative bacteria, which often have different susceptibilities to antibiotics, aiding in the selection of an appropriate antibiotic treatment. Next, going to start the MCQ. Question: what is the primary stain used in gram staining? Option: A. Safranin, B. Crystal violet, C. Methylene blue, D. Malachite green. Answer: B. Crystal violet. Question: which of the following bacteria would appear purple after gram staining? Option: A. Escherichia coli, B. Staphylococcus aureus, C. Pseudomonas aeruginosa, D. Neisseria gonorrhoeae. Answer: B. Staphylococcus aureus. Question: what is the role of iodine in the gram staining process? Option: A. Decolorizer, B. Primary stain, C. Mordant, D. Counterstain. Answer: C. Mordant. Question: which step in gram staining differentiates between gram-positive and gram-negative bacteria? Option A. Application of iodine, B. Decolorization with alcohol or acetone, C. Application of safranin, D. Application of crystal violet. Answer: B. Decolorization with alcohol or acetone. Question: gram-negative bacteria appear what color after gram staining? Option: A. Purple, B. Blue, C. Pink or red, D. Green. Answer: C. Pink or red. Question: which component of the bacterial cell wall retains the crystal violet stain in gram-positive bacteria? Option: A. Lipopolysaccharide layer, B. Peptidoglycan layer, C. Outer membrane, D. Capsule. Answer: B. Peptidoglycan layer. Question: what is the counterstain used in gram staining? Option A. Methylene blue, B. Safranin, C. Crystal violet, D. Iodine. Answer: B. Safranin. Question: why might a gram-positive bacterium appear gram-negative under the microscope? Option: A. Over-decolorization, B. Under-decolorization, C. Use of wrong stain, D. Presence of capsule. Answer: A. Over-decolorization. Question: which of the following is a characteristic of gram-negative bacteria? Option: A. Thick peptidoglycan layer, B. No outer membrane, C. Thin peptidoglycan layer and presence of an outer membrane, D. Retain crystal violet stain. Answer: C. Thin peptidoglycan layer and presence of an outer membrane. Question: which bacterium is most likely to be gram-positive? Option: A. Escherichia coli, B. Streptococcus pneumoniae, C. Neisseria meningitis, D. Salmonella typhi. Answer: B. Streptococcus pneumoniae. Question: negative bacteria appear pink or red because Option: A. They retain the crystal violet stain, B. They have a thick peptidoglycan layer, C. They take up the safranin counterstain, D. They do not react with iodine. Answer: C. They take up the safranin counterstain. Question: which of the following correctly describes gram-positive bacteria? Option A. They have a thin peptidoglycan layer. B. They appear pink or red after gram staining. C. They have an outer membrane. D. They retain the crystal violet stain. Answer: D. They retain the crystal violet stain. Question: gram staining is particularly useful in identifying bacteria based on Option A. Their metabolic activities. B. Their ability to form spores. C. The structure of their cell walls. D. Their motility. Answer: C. The structure of their cell walls. Question: which of the following statements about gram-negative bacteria is true? Option A. They lack an outer membrane. B. They have a thick peptidoglycan layer. C. They are typically more resistant to antibiotics than gram-positive bacteria. D. They retain the crystal violet stain. Answer: C. They are typically more resistant to antibiotics than gram-positive bacteria. Question: which of the following bacteria would likely appear gram-negative? Option A. Staphylococcus aureus. B. Escherichia coli. C. Bacillus subtilis. D. Streptococcus pneumoniae. Answer: B. Escherichia coli. Next, but not in the list. Note: if you perform the gram staining steps in reverse order, the results will be inaccurate and misleading. 1. Counter staining with safranin first. If you start with safranin, all the bacteria will take up the pink-red stain. This will prevent you from correctly differentiating between gram-positive and gram-negative bacteria, because both types will initially appear pink. 2. Decolorization with alcohol or acetone. Next, decolorizing after safranin will remove the safranin stain from gram-negative bacteria, but might not fully remove it from gram-positive bacteria, especially since the safranin was not bound tightly to the peptidoglycan layer. This could lead to confusion, as some bacteria may lose their color entirely or retain a faint pink hue, making interpretation difficult. 3. Adding iodine, mordant, after decolorization. Adding iodine at this stage will not form a crystal violet iodine complex because the crystal violet has not yet been applied. The iodine will not have any effect, and it will not bind to the cell wall. 4. Applying crystal violet last. Finally, if you apply crystal violet at the end, both gram-positive and gram-negative bacteria will take up the violet stain because there was no iodine treatment to form the complex, or a proper decolorization step to differentiate the bacteria. All bacteria will likely appear purple. Conclusion: reversing the steps will result in all bacteria appearing purple or pink, preventing you from distinguishing between gram-positive and gram-negative bacteria. The staining process relies on the correct order to ensure accurate differentiation. I hope you enjoyed the session. If you have any doubts, comment below. See you in the next video.